I think it's gonna be really helpful. So that's how I'm gonna break it up. The other thing, the the exam really was an improvement. Now I want to start with the good news, and I know that it's possible that not everybody um, improved the way that they wanted to, and it could be that you're still not doing as well as you want to in the class. But I did want to encourage everybody that I saw a lot of improvement um, as a class. The class average on the exams, and I talked about this a little bit a few weeks ago after the first exam. I don't usually talk about the class averages because a lot of the times the average includes, people who might not have taken the test and things like these zeros are included. There's a lot of weird statistics that are part of it. And I also don't really like to report the class average too much because my take on that, my thought, it's just me personally, is that I don't think that the class average matters because the exams are established by the department, uh, and what matters is you as an individual, if you're improving and doing as well as you want to be doing individually. There's no curve, and there's no scaling of the grades or anything like that. There's no extra credit that I can give. That's just a part of being a part of this department at Gwinnett Tech. These are rules that we all go by when we teach. Each individual instructor isn't able to do any that stuff like that. So for that reason, I try to focus on. I'll get to that. I try to focus on each individual student's, in performance when I look at it. So what I saw overall was like a 10% improvement, which was a really, really drastic improvement overall. Again, I know that may not be you. You might not have been. You might still not be where you want to be. Yeah, you can't see the exam. So the overall improvement that I saw was really better than I've seen most semesters from exam 1 to exam 2. Now here's why that's a big deal, and here's what answers to Shai's question. I hope I pronounced your name correctly. The reason why I kind of skipped over your question: no, the lowest grade will not be dropped. All right, so again, we talked about this at the beginning of the semester. The lowest grade will not be dropped. What will happen is if you score higher on the final than your first exam, that higher grade on your final will count both as your final and will replace your first exam grade. So again, like I said, I don't want to spend too much time talking about it, but I do want to make sure that everybody understands the policy that we kind of have. And this is only for A&P 1, by the way. I'm also teaching in a 2 class this semester, and we don't do this in A&P 2. So let's use some figurative numbers. Let's play with some numbers. And let's say we have a student that made an 85. Let's see, can everybody see that okay? Let's flip to this one. Sometimes the screen is just really, really crazy at the first of class, the beginning. So let's say student made an 85 on exam 1. Here's exam 1, here's exam 2, 3, 4, and we'll put F for the final. They made an 85, and they've improved. They got a 90, they got a 90, 92, not a 29, 92, an 89, and this 85 is kind of pulling the average down a little bit, and but on the, you can see that there's some overall improvement. That it's kind of coming up a little bit, and there's improvement. And then on the final, they get a 92, right? It's fully comprehensive. So the 92 counts as the final, but then what I do in Blackboard is I replace the 85 with a 92, right? So the reason why we do this, and again, like I said, we don't do this at all in A&P 2. They don't do it that I know of. I don't think they do it in I think that it's just kind of one of these things that we do to help students that are new to our courses, give them some encouragement, and I hope that it does. I hope this gives you some hope that you can continue improving. Yes. Okay. I felt like I was clear. Thanks, Joshua. Felt like it's pretty clear on that. It does not replace the lowest, it only replaces the first. So maybe I should, how about I do it this way? How about I do it like this? Look, 85, 90, 82, 89, 92. How about that? How about that? All right. Here's what will happen if this is the case. Is everybody tracking me on this one? Watch. Still only replaces the first, and only if the first is lowest. So if you don't do as well on the final than the first, you get to keep the first that's higher. It'll stay. So if you got an 82 on the final, then your grades, nothing would change. Your first exam 85 would stay. And these do not get changed. Exam 2, 3, and 4 do not get changed. Did I explain that better that time? You were multitasking us on you. I mean, this is the problem, guys, with going to class online when you are sometimes at home or maybe at a library or a coffee shop or something like that. Multitasking kind of just has to happen, especially if you're at home and you're a parent and the kids are around. So that's completely understandable. So hopefully I was able to kind of make that a little more clear about what that policy is. And it's something we do that just encourages students to continue to push on and improve. So that's why I'm kind of praising you guys, a group, because I really felt like there was good improvement on this second exam overall. And again, I know that not everybody did, but I want, even if you didn't improve from exam 1 to exam 2, remember that from exam 2 to exam 3 is completely new, different material every time, and that you can continue to keep trying and improving. If you're not improving, then you definitely want to talk to one of maybe your program advisor or something about what options are and things are like that. As we get a little closer to the withdrawal date, which is I think March 22nd for this semester. But if you got your mind on the future and you're still thinking ahead, you're moving ahead, remember we talked about this at the very beginning, and I'll remind everybody again, I want to keep this in your mind. The final exam review is always there. It's in the back of the book. And so for the material we started last week, that will be material for exam 3, that's also in the exam review, starts at the top of page 229, right there on 229. Okay, so you are, for exam 3, you have an exam 3 review in the middle of the book, starting on page 141, okay? But the final exam review, I'll just kind of write it like that covers exam 3 material, okay? Why not try to double up? It's you're studying the exact same material. Why not look at the stuff you need to know from this material for the final? And it starts on page 229, okay? I know I didn't remind you about this as much as I should have for exam 2, but I did talk about it on the first day of class back in January. But man, there was a lot going on. So I apologize if you feel like I'm being redundant and you guys are back there rolling your eyes saying, why does he keep saying the same thing over again? Because stuff gets busy and stuff gets crazy and it's easy to forget. So from page 229 over to page 231. So it's question 20 over to question 30. Question 20, on pages 229, it's question 20 to question 30. That's the exam 3 material. So, you're learning the material. So we're on page 103 today, for example. We're starting chapter 10. We're going to go through chapter 10, right? After I get through all this preamble. So we're going through chapter 10 today. But as you're studying your lecture notes in chapter 10, also be looking at exam 3 review material over here, page 140. I don't know exactly where it is here we go, page 143. Doubles up, page 143. And then somewhere over there in the final exam review, it's all the same stuff, but you can be studying for exam 3 while you also got your mind on dominating the final exam, which is totally just review stuff. It's all old stuff. It's all comprehensive stuff you've already been tested on. So I think it just would be so much better use of your time. Okay, let's go. I'm ready. You can tell I'm ready. This stuff is really complicated, but it's some of my favorite stuff to study about, to talk about, to teach about, because it's amazing. It's just amazing to me. What we're talking about today about how muscles contract is not just how you move your skeleton and move your body. A lot of it is how it's like you blink your eyes. As fast as your eyes can blink is how fast all this stuff happens. So there's right at the top of page 103, 3 kinds of muscle tissue. They're listed in that table. Fill out the details, as many of these details as you possibly can. So let's go through that really quickly. Our focus is going to be on the far right column. Our focus is going to be on skeletal muscle in this chapter, but there's also cardiac muscle and smooth muscle. So generally for cardiac muscle and smooth muscle, it's mostly 1 nucleus per cell. So you might have, your table might be kind of like this. Cardiac, smooth, skeletal. In that first row, you've got one. Sometimes cardiac muscle has 2 nuclei per cell, but nothing skeletal. Hundreds or even thousands of nuclei per cell. All right, so big contrast with these cells that we're going to be looking at. Location of nuclei, middle, center, whatever, however you want to say it. The center of the cell, the middle of the cell, however you want to do that. And then, sorry about my handwriting there, peripheral. So the nuclei of skeletal muscle cells, the nucleus is out at right under the plasma membrane, right under the cell membrane. It's not in the middle of the cell like it is with a lot of cells. Striations, yes. No. And yes, kind of put a Y for yes. Cardiac muscle is striated. That means when you look at it under the microscope, it has these little alternating light and dark bands. Skeletal muscle is. Cardiac muscle is. Guess why smooth muscle is called smooth muscle? Because it doesn't have those bands. It was named smooth muscle because it doesn't have those alternating light and dark bands. Intercalated discs, yes. Cardiac muscle is the only one that has those intercalated discs, which are the places where the cardiac muscles attach to each other. Voluntary, definitely no. You do not want to be in control of your heart. What would happen to your heart if cardiac muscle was voluntary? What would happen when you fall asleep? It would relax. You would die. This is not something you want. Right, yeah, right. The one that is voluntary, and this is going to kind of be a big topic that we're going to talk about today in the second half of class, is what it means for skeletal muscle to be voluntary. Cardiac muscle, not voluntary. Smooth muscle that drives your food through your digestive tract. No, again, you don't want that to be you don't want to have to think about moving the food from your stomach into your small intestine and then from your small intestine into your large intestine. It's just not anything. You got more things to do with your life than to think about that. The shape of the cells, I think the best way to kind of describe cardiac muscle cells is branched. That's unusual. Smooth muscle cells are often described as spindle shaped, which is a word that not everybody knows, but this, like that, with a nucleus in the middle. I've heard it described as tapered on either end, kind of pointed at each end, and the cells can be really long. And skeletal muscle, I don't even know if I spelled. Not only can you not read it, I probably didn't even spell it right. Cylindrical. Cylinder. Long, round, like an unsharpened pencil. Is that a good analogy? An unsharpened pencil? Like a brand new wooden pencil before you put in the sharpener? That's what skeletal muscle cells are like. They're not branched. They don't taper at the tips. They don't get smaller at the ends. So they're long and cylindrical, like rods. Okay. Characteristics of muscle tissue. So I kind of got this from the muscles in threes. I kind of got this from Dr. Sadri. So I got to give her credit for kind of helping us think about the characteristics and functions of muscle. All the stuff at the bottom of 103 and 104, and kind of summarizing it together in these clusters of three. So there's three types of muscle, which we talked about in that table there. Three properties of muscle, which is at the bottom of 103. Page 103. Excitability, that means it's responsive. That means it can be excited. Contractility, which means it contracts, it shortens. It shortens when it's activated, when it gets excited. And elasticity means it snaps back when it gets stretched. So it's stretchable and it can snap back. And a number of different functions of muscle, like movement. Now for skeletal muscle, movement of the skeleton. Okay. But movement of food, if we're talking about smooth muscle. Movement of blood, if we're talking about cardiac muscle. The main job of cardiac muscle is to pump blood. So all kinds of movement related to muscle. So not just, again, our focus is going to be skeletal muscle, but not just skeletal muscle, movement and bone. Maintaining posture. This is mostly skeletal muscle that helps you maintain your posture, which is not movement. That's like if you're just sitting up and sitting still, you're still using skeletal muscles. They're just holding a contraction. They're sustaining a contraction to keep your posture. So that's a little bit different from movement. And also body temperature. A lot of your body temperature a lot of your body heat comes from skeletal muscle. Why does skeletal muscle increase body temperature? Because it converts a lot of glucose into ATP, and that releases a lot of energy. And so that process we'll talk about later is called cellular respiration, and it generates a lot of energy. How does skeletal muscle aid in homeostasis of body temperature? Well, it helps warm you up when you're cold. And in fact, what does skeletal muscle do when you're cold? What does it do? It shivers. Yeah, that's what shivering actually is. Shiver is repeated muscle contractions of skeletal muscle. So since skeletal muscle can generate heat by both friction and doing that process of turning glucose into ATP called cellular respiration, it can generate a good bit of heat. So shivering is the body's response to that drop in temperature. Let's talk about a little bit about muscle anatomy. A single muscle, is it a tissue, an organ, or a system? Well, I'll give you a clue. The whole system is called the muscular system. So a single muscle is an organ. Yeah, it's an organ of that muscular system. So each individual muscle that you're learning or will be learning the names of in lab mostly is where you'll be focusing on learning all the muscle names. So an example of one that a lot of people might be familiar with, right here on the shoulder. It's a common place to give a shot, to give a vaccination. It's not the only place, but it's a common place. That muscle is called the deltoid, and typically we'll give the shot right over here on the side of the shoulder, right in that part of the deltoid. So that's the deltoid muscle. It is a single organ of the muscular system. Right over here on the front of the arm, right there, is another one that is pretty well known, biceps brachii. As a former adolescent teen boy, that was a big focus of working out at the gym back in the day, when we thought that was the way that we could get girls to look at us, which was a silly, stupid thing. But that's why most of us grow up kind of knowing the name of the biceps brachii. But on the backside, it's the triceps, right? The triceps muscle. And each one of these is an individual organ. So remember that as you're learning all these names in lab, each one of these has an individual name, is an individual organ of the whole thing. And that whole single organ is surrounded by a layer of tissue that kind of holds it all together. It's labeled on this diagram right here called epimysium. Epimysium. And it surrounds the whole muscle, the entire whole muscle. You can see it on this image right here, sort of cut and peeled away, so you can see it's a layer of tissue that surrounds the whole organ. Now, let me make a note of this for you, because this is one of the places where I feel like it starts to get a bit confusing. Over on page 104, it talks about the layers that you need to get through to get to muscle. To reach skeletal muscle, well, where is skeletal muscle located? It's deep to the skin. To reach skeletal muscle, the following layers would have to be removed: skin, epidermis and dermis, hypodermis, which is not part of the skin, all right? Then the deep fascia, and the deep fascia is usually found around groups of muscle, sometimes around a whole set of muscle of the arm, for example. Let me erase this and kind of redraw. We might see like a deep fascia under the skin, but wrapping all the way around these muscles of the arm. Okay, and it's just another layer of it's almost like a tight sock on the arm is this deep fascia, but it's made of dense, irregular connective tissue. What else surrounding bone is made of dense, irregular connective tissue? Dense, irregular connective tissue around bone. Around bone? The name means around bone. Yeah, periosteum. Periosteum. Right, I know you guys see the word tendon here on the screen. Tanya said, I accidentally spelled periosteum periosteum. Is that right? Misspelled it. I get you. I feel you. All right. Okay. But here's the thing. So you guys are not far off. I was talking about the periosteum. Okay. So I was talking about the periosteum is around bone. The epimysium around muscle is made of the same kind of tissue. And the in-between that connects epimysium to periosteum is a tendon. The difference is a tendon is not dense irregular, a tendon is dense regular connective tissue. But they all blend together. So the epimysium, the deep fascia, kind of merge together, the epimysium and the deep fascia. They sort of merge together here at the end of the muscle to become a tendon. And then the tendon, the fibers of the tendon spread out and interweave with the I'm showing it interweaving with the periosteum to attach the muscle to the bone. So that's what the periosteum is. The fibrous layer of the periosteum's function is to allow for attachment of tendons and ligaments. All right? So we're not getting far away from what we've just been talking about with exam 2 material on the bone and the joints. So when we peel this epimysium away, I wanted to mention that about the deep fascia, because the deep fascia, it says there, the deep fascia can surround an entire muscle, but it can also surround whole groups of muscles. And it's not the same as the epimysium. The epimysium surrounds one single muscle. The deep fascia can also surround that one muscle, but in some places in the body, it surrounds whole groups of muscles, like we see on the arm and the leg. But in either case, if you were to try to get to the muscle, you'd have to go through skin, hypodermis, and then deep fascia. Okay? A tendon, muscles connect to a bone via a tendon. Name the tissue I just did, dense, regular connective tissue. A tendon sheath we talked about last week, and its purpose is to reduce friction. It surrounds a tendon. It's basically a synovial pouch that surrounds a tendon. So we're now kind of moving on. I'll talk about aponeurosis later, but now we're moving on to page 105. And the entire muscle is an organ, because it's an individual organ of the muscular system. I don't want to get too much into the classification of it, but that question why at the very top of page 105 is it has its own unique individual blood supply. So that's one of the things that anatomists do when they classify each of these muscles. They say this one is a separate organ because it has an artery going to it, but it's not shared blood supply. But the other thing is, remember what makes an organ is it has to be composed of at least two tissues. So the entire muscle is surrounded by a connective tissue called, right here, epimysium. Epimysium. I'll go back to my previous slide where I had it written a little bit bigger over here, right here. One muscle is a single organ surrounded by epimysium, and then the entire muscle is kind of made up of these individual pieces called fascicles. So if you see peeling the epimysium away, what you see underneath now is actually a collection of these other tubular or cylindrical structures that are called fascicles, and that's what's pulled out right here. That single thing is a single fascicle. It's a bundle of smaller things. So you can see this is a close-up view of it here. So this is a fascicle. And now the fascicle is surrounded by a connective tissue. This is your second blank at the top of 105. It's surrounded by a connective tissue called perimysium. Perimysium. Okay? So you can see here's a whole muscle. It's a collection of fascicles. 1, 2, 3, 4, 5 little fascicles, 6 and 7 and so on. One of them is kind of pulled out, and then we do a little cross section so we can see it up close. And what's surrounding it is the perimysium. But it also is made of a bundle as well. It's a bundle of things together, and it's a bundle of muscle fibers. Each one of these is an individual muscle fiber. That's our word for muscle cells. And each individual muscle fiber is also surrounded itself by an additional connective tissue layer called endomysium. Endomysium. All right, so again, top page 105, a muscle fascicle is a group of muscle cells. One muscle cell is also called a muscle fiber. That's what we're going to call it most of the time. Muscle cells or muscle fibers contain long cylinders called myofibrils and are surrounded by connective tissue called endomysium. So that should get your blanks filled in there at the top of 105. Or if you've already filled them in, that just helps you double check that you got them right and that you're studying the right stuff. Now we're going to start taking a look at these other things called myofibrils and myofilaments. Myofibrils and myofilaments. To do that, we're going to start a closer look at an individual muscle fiber. An individual muscle fiber. Because an individual fiber, if you look at it from the end right there, like this, you see it's a bundle of little things too. All right? And you can already start to see how complicated the organization of a single muscle is. It's bundles of bundles of bundles. It's like, hold on. Sometimes it helps when I'm out here in the laundry/storage room. Hang on a sec, I got something I didn't think about for a minute. I don't know if you guys can see this. Okay, down here on my camera view. I hope you can. So, when you homeschool, you always got to have lots of pencils around. So just buy bundles of pencil boxes off Amazon. But it's kind of like if this is sort of my 3 different pencil boxes are one muscle, and my hands are the epimysium. I don't know if this is going to work. We're going to see. I'm making this up as I go along. All right, one muscle surrounded by epimysium. All right, I hope you can see that. Okay. Now this muscle I have, this one muscle, is composed of 3 subparts. Each of these is a fascicle. Each one of these is a fascicle. Okay, so I'm going to separate one of these out, just like they do on the picture. Like that. Okay, there it is, fascicle. And the cardboard of the box itself is the perimysium that surrounds a single fascicle. Okay? So the cardboard is the perimysium. Yeah, let's do it that way. Okay, but look when I look inside my fascicle, I can see that my fascicle is actually made of individual parts too. Let's pull one of those out. Look at that. My whole fascicle, surrounded by perimysium, is actually made of these individual things too. And each one of these is a muscle fiber, a muscle cell. And the paint on the pencil is like the endomysium, the connective tissue that surrounds each one. And it actually helps, just like the pencils, it keeps them separate and helps them slide past each other separately, because when muscle fibers contract, sometimes they need to contract independently of the others. Hopefully you guys can see what I'm trying to illustrate. This one can contract independently of others, and it's actually the endomysium that sort of keeps them separated so that one can contract without disturbing the next door neighbors. But they're all collected in one grouping called a fascicle, which is a cluster or collection of muscle fibers. And that grouping is surrounded by perimysium, which is represented by my cardboard. I'm having trouble making sure I'm aiming at the camera. Okay? And that's one fascicle surrounded by perimysium, but the whole muscle is multiple fascicles surrounded by epimysium. Pencil dust all over my desk now. good. I hope that helped. It's crazy. It's crazy, you guys, because what I just showed you was like, take a look down here in the bottom right. This would be like one of my single pencils, just one of the pencils. But now we're going to zoom in to the microscopic level of the pencil itself. Hold it up to the camera and see how it looks. I don't know if it's going to be blurry. We're going to zoom in. No, it's blurry. We're zooming right in to look at the structure of the inside of the individual muscle fiber. And we see that there's stuff in there too, and it's cylindrical. It's bundles. And they all have names. Why did what work so well? I don't know if it worked well at all. If it helped you learn it, then it worked. That's all that matters. It's never a bad idea to keep a few boxes full of pencils hanging around. So check this out. Here's another view. So epimysium covers the whole biceps, yes, or the whole deltoid, yes, or the whole triceps, yes, yes. Then perimysium covers groups of, or known as fascicles, within the biceps, yes. And then endomysium covers individual muscle fibers within the fascicle, yes. Yeah. So Tanya, what you typed there is correct, but I just took it a step further. Yes. Yeah, so here's a different illustration from the same textbook. Here's our whole muscle, a whole skeletal muscle surrounded by epimysium. Remember, this is not the same. Good. Some of you feel like you're getting it. Good. This is not the same as deep fascia, which I'm going to kind of draw in blue up here in the upper left. This muscle may also have deep fascia that surrounds it and sometimes maybe another nearby muscle. There might be another muscle over here. Okay? So we got deep fascia and then the endomysium. Ah, crap. Sorry. Bad. The epimysium. So I correct myself. Take my own advice and check myself before I wreck myself. So the epimysium surrounding the, as Tanya typed in there, the epimysium that covers the biceps. So let's play like this is the biceps, all right? So the epimysium surrounds the outside of it, there. Maybe this is the deep fascia. Oh, yeah. That's what this is trying to be. They've got the word epimysium on here, but this thing right out here is actually the deep fascia. Sorry, guys. This is not that big of a deal. It's just the way the illustrator chose to illustrate it. I just want to make sure I don't confuse you even worse in trying to make this make sense. So this is really the epimysium, this more like bluish purple here. And each of these things here is a fascicle. It's a fascicle. And here's one fascicle slid out, covered in perimysium. And now we're going to take a closer look at this fascicle here. So here's the perimysium of that fascicle. All right, this is kind of what they think it might look like without that perimysium all the way around it. And perimysium is a bundle of fibers, muscle fibers. And here's a single one of those muscle fibers kind of slid out, like sliding a single pencil out of the box. And here's one of them here, and we see that it actually contains these little cylindrical things inside the cell. There's a nucleus, these little things are mitochondria, and these individual things are called myofibrils. And these are long, they run the whole, these aren't little chunks like they look up here in the upper right. These are long things that run the whole length of the cell. And that's a myofibril. The deepest, Heba, the deepest connective tissue is endomysium, yes. The deepest connective tissue. That surrounds a single muscle fiber. But I want to make sure you guys know I'm going deeper than one cell now because we're looking into a single muscle cell, a single muscle fiber that also contains these cylindrical organelles called myofibrils. And they look like this up here. This is kind of the zoomed in view there. I couldn't get it all on the same column like it's in your textbook. So I had to kind of stagger it side by side. So this is one myofibril. And then check it out. Surrounded by sarcoplasmic reticulum, which is a special type of endoplasmic reticulum. But then a single myofibril is actually made of bundles of what are called myofilaments that come in two varieties, thick and thin. Thick and thin myofilaments. Thick and thin myofilaments. All right, so a muscle is a bundle of fascicles. A muscle is surrounded by epimysium, and that epimysium holds all the fascicles together. A fascicle is a bundle of muscle fibers. That fascicle is surrounded by perimysium that holds all the muscle fibers together. A muscle fiber is a single muscle cell that contains bundles called myofibrils. It's a bundle of myofibrils and other cellular stuff. The muscle fiber is surrounded by endomysium. A single myofibril is actually a bundle of myofilaments. So of all these structures, the tiniest one, the smallest one, is the myofilament. The one that's at the kind of the tiniest microscopic level. Okay. Let me see. Got next. Here we go. All right. Hopefully this kind of doesn't add too much or take away too much. This just to sort of clarify. So this is a different view of the same, kind of a different view of the same cell, but from a different angle. We're kind of switching it around. So this is a single muscle fiber. It doesn't have any of the endomysium around it, but if I just kind of draw something like this, it's a loose connective tissue, kind of like an areolar connective tissue that would surround this one muscle fiber. That would be the endomysium. Okay. And now we're going to zoom into this area right here where we have a little chunk or a section of the cell. This on the outside surface is called the sarcolemma. It's the plasma membrane, the cell membrane. Oh, there, it's labeled right there, sarcolemma. And it's composed of these myofibrils, which is a single one of these things right here. They always kind of remind me of, I'm not a smoker and I don't want to promote it, but if you can imagine somebody kind of shaking a single cigarette out of the cigarette pack and it kind of sliding out, that's what that sort of reminds me of. It's like pulling a single one of them out of the pack, kind of slides out, or pulling a single pencil out of a box. That's a myofibril. But it actually consists of two types of myofilaments, called the thin filament and the thick filaments, and there's lots of them. The thick filaments are mostly composed of a protein called actin. Sorry, thick filaments are composed of myosin. I was looking at the word actin. The thin filaments actually consist of three proteins: actin, tropomyosin, and troponin. Okay, we'll take a closer look at those in a second as well. All right. The sarcoplasm is basically the term for the cytoplasm, or the cytosol, but it contains lots of mitochondria. And I think that probably makes a lot of sense. There are a lot of mitochondria in the sarcoplasm, what it says, what organelles would you expect to find in abundance in the middle of page 105? Mitochondria, lots of them. T-tubule, what is it? Well, we'll talk more about it later. I'll just kind of point it out. They start at these little holes right out on the surface of the cell. That's why these cells have a special name for their plasma membrane, because their plasma membrane actually has these little holes that drop down into the cell and they weave around the myofibrils. So these things that are shown in yellow are actually tubes that start at the surface of the cell. It's like I imagine it's kind of like a manhole that you'd see on a city street. So if like you were, if you were walking along a street, that's you walking along a street, looking, down inside of a manhole, it leads to little tunnels, like little subterranean tunnels below the street. And that's what that T-tubule is. And another major thing is sarcoplasmic reticulum. And what does the SR store? This is a little fill in the blank there in the bottom middle of page 105. And SR is going to be critical. And it is our calcium storage. Yeah, calcium. Calcium 2+. Calcium plus. That's right. Yeah, this is where calcium is stored. Let's see, it says draw a muscle cell. So I cheated. Label the sarcoplasm, the sarcolemma, the T-tubule, and the sarcoplasmic reticulum. So I cheated and just drew you a picture. I cheated and put a picture from the textbook. But this is basically, there I drew you a muscle cell, guys. You're welcome. But it says draw it cut in cross section showing the myofibrils, the sarcoplasm, the sarcolemma, and the T-tubules. That one I think would maybe be a little bit, I failed. I know. It's because I'm such a terrible artist. So thank goodness your grade doesn't depend on your artwork, right? So if I drew it in cross section, then I would kind of say, this is the sarcolemma right out here. That would be, I'm going to circle it. Again, I'm going to cheat from the diagram. There's my sarcolemma. And then I would just draw a bunch of circles in there. And those are my myofibrils. And then the space between them is the sarcoplasm. The sarcoplasm is kind of the space between the myofibrils. The myofibrils would be my circles, like that. And then there would be T-tubules kind of weaving in between. Let me draw them a different color so they stand out. T-tubules would be kind of weaving all around in between, wrapping around those. And that I would just do the rest like that. Sorry, guys. Sort of embarrassed about my tragic artwork. But your artwork doesn't matter either. So if you feel self-conscious about your drawings at the bottom of page 105, it's a practice. It's a good thing to kind of practice because it helps you personally see that you have a good understanding of the arrangement of these parts. And you know who does, I hate to keep turning it back over to Dr. Schultz, but she does a really great drawing. She does many great drawings of many things with her little dry erase markers and her whiteboard. And every time I watch them, I'm impressed. I'm like, man, dang, Catherine, good. Lots of practice. She's had years and years practice. Yeah, she does a really great job of that. And if you want to just copy her, that's all good. Don't copy me. What makes up myofibrils and myofilaments? Let's see. I wanted to skip those and come back to these. Oh, here's a good question. Let me give you some. Don't answer just yet. Give everybody a few seconds to read the question, okay? Don't put anything in the chat yet. This is from I think from an old quiz. So this is maybe potentially a question that might be on the upcoming test on exam 3. But this is the kind of question that you might see. All right, so anybody want to go ahead and put an answer in the chat real quick, A, B, C, D, or E? Yeah, C. So C is the only one that's smaller than a myofilament. Larger, 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 okay? However, think about this. This is, whenever I put practice questions up like this, think about how might we change this question a little bit. You know, and now if I change it, admittedly, we might have more than one correct answer because I would have to tweak the answer choices. But I think, obviously, we wouldn't choose fascicle. It's not going to be smaller than a fascicle. Still myofilament, but also a muscle fiber as well. Okay, so a muscle fiber makes up a fascicle, and a myofilament makes up a muscle fiber. So both of those would be smaller than a fascicle. Now, again, like I said, to make it a question on one of our exams, we would need it to have one correct answer. So we'd have to tweak the answer choices. But I want you guys to see how, you know, see behind the scenes about how us teachers, the faculty, all write questions like this, and we kind of write a question, and then you see how we can just change a little bit and make it a completely different question about the same concept or the same idea. But I want you guys to also be thinking, how would this be tested on the test? How could I write a multiple choice question from this information? Okay. You know, how about, well, I'll show you some examples later. I don't want to waste any time on that just now, but you know, it's a good idea to think about, if I was the teacher trying to explain an actin thin myofilament, could I explain the basic structure? But then, how would I know if my students understood the basic structure of a thin filament? I could say something like, thin filaments contain all of the following except, something like that. Because thin filaments actually consist of a bunch of different kinds of proteins, as we'll see on the next slide. Let me for now talk about thick filaments, but I want you to see how, as you're studying, you can actually be thinking, if I was the teacher, how would I write a multiple choice question? Now, you're not the teacher and you're not writing multiple choice questions, but you know what you do when you start thinking like that? You start thinking like the teacher. And as a teacher, I'll promise you that's not a bad thing. It's a good thing. If you can start thinking like us, you're coming over to our side. Come over to the dark side. We've got cookies. Okay, so the thick filaments, it's kind of at the bottom of page 106. We'll look at this, like, let me go back to this sarcomere thing that I had over here. I skipped that. When you look at a muscle, when you look at a whole muscle fiber from a lower magnification in a microscope, that's where the name striated comes from. And look at all those nuclei all over the place. You can see all the nuclei are all kind of tucked right up under there. Oh yeah, I like snickerdoodles too. Sure. Yeah. I mean, if you guys come over to the dark side, look, the point about the dark side is you get whatever kind of cookies you want. You also have to sell your soul. But no big deal. Hey, right? But as long as there's cookies. Just kidding. So all these nuclei, right, they're not really inside the cell. They're all pushed out against the edge. You can see these peripheral nuclei and the striations. What gives it the striations? The distance from band to band is often known as a sarcomere. And that's kind of, I didn't want to totally skip it, but I'm going to talk about it more here in a minute. The screaming going on. Ow, wow. Okay, I'm not in charge. I'm not on dad duty right now. So the sarcomere, these alternating light and dark bands, are made of these thick and thin filaments. So if you don't mind, for now, the stuff that's in the middle of 106, I'm going to kind of skip it and then back up to it, because I want to talk just about the thick filaments first. And if I was to sketch a thick filament, I'd kind of sketch it like this, like that, and then I'd kind of do another one of those, and then do another one like that, and then kind of do another one like that, and that's probably about as far as I'd go, and I'd say, okay, that's fine. But I would remind myself that that first thing that I drew, I'm going to kind of pull that one thing out, sort of like that. That's a single protein that makes up the whole thick filament called myosin. It's like a single piece of myosin. The thing that we call a thick filament is a bundle of myosins. You can probably see that bundle a little bit better down here. That's a bundle of myosins. And their little things that stick out are referred to as the myosin heads. So it says this kind of at the top of page 107. A thick myofilament contains many myosin proteins. Sketch the myosin protein shaped like a golf club. Mine's not really shaped like a golf club, but it looks more like a sideways musical note or something, like a little eighth note. Okay, so if you sketch one on your page, you can make it go just like this, and then a little loopy at the end like that, and you're good. And this is the part that's called the head. And the importance about the head is the head can, now this is listed on page 107, the head can break down ATP, the head can pivot, which means it can kind of wiggle back and forth, like that. You can see it, there's a better illustration. You can see it kind of rocking back and forth, like that. It can pivot, and it can also bind to the other protein, which is called actin, the thin myofilaments called actin. Okay? So that's a single myosin bundle together called a thick filament. But I want you to see how these are arranged over here, alternating in between the thin filaments, which are the red ones. The thin filaments are the little red ones, and the thicker filaments are the purple ones that kind of go back and forth. If I understand correctly, does it break down ATP to become ATPase? No. It has the enzyme. No. When you break down ATP, I'm going to put the question back on you, and Trisika, you don't have to answer it by yourself. I'll just kind of put it back to the rest of the class, and we'll see if everybody's listening. When you break down ATP, what do you break it down into? Let's see who can type this one the fastest. Yeah, there you go. ADP. Look, they wanted to type it so fast, they didn't even bother capitalizing it. They're just like, I know this one. All right, so, it's a good question. When you break down ATP, you get ADP. Yes, Heba, the ATPase is the enzyme that breaks down ATP into ADP. And all that, and are you asking, is it in here? Or does she say something about it in the video? Because I can't recall that it's right here in our lecture notes. But this is the phrase. This is the phrase. We say, awesome, good, she says it. Now, remember, you don't have to know it if it's not in the lecture notes. That's kind of usually my policy. But here's the deal. She is saying, accurately, that the myosin head has ATPase-like activity. That's the molecular biologist phrase in me. The myosin head has an enzymatic active site that can turn ATP into ADP. Therefore, we say the myosin head has ATPase activity. It has ATPase-like activity. So the head can act like ATPase and turn ATP into ADP. Does that confuse it or clear it up? Confuse it, clear it up. Okay. You said from the video that was confusing in the video. Yeah, but did I fix it? Okay. She's not answering me, Tristica specifically. I was asking you if I cleared it up, but that's all right. You can leave me hanging. That's okay. I see how you are. That's fine. Thin filaments. I'm going to move on with my life. Thin filaments. The thin filaments consist of, as I already said, the thin filaments actually consist of 3 proteins, whereas the thick filaments are really mostly just myosin. And I say mostly because there's others, but you just need to know myosin. Thin filaments consist of 3. 1, 2, 3. I've got them listed here. And they consist of actin. And actin is where myosin actually attaches. Okay, so on our previous slide, we said one of the things the myosin head can do is it's got ATPase activity, it can flip back and forth, but it can also attach to actin, right? So these little pockets that are in the kind of bottom right-hand corner, if you look at the filament up close, there's these little yellow spots that myosin can attach to, but see, it's labeled active site, but it's covered. It's covered by this other protein called tropomyosin, which wraps around actin and covers those active sites. Okay? So tropomyosin actually blocks myosin from attaching to actin. Even though myosin wants to, it blocks it. So when tropomyosin is in place here, myosin cannot attach. But there's somebody attached to tropomyosin that can control tropomyosin, and it's called troponin. It's known as troponin. Troponin attaches to tropomyosin, and troponin also has a calcium binding site. Okay? Now remember, you can answer this in 2 letters, but I want you for the test to make sure you know what these 2 letters stand for. Where in the cell, in the muscle cell, is calcium stored? Where in the cell is calcium stored? You can answer with just 2-letter abbreviation, but I want you to make sure you know what it stands for the exam. Where is calcium stored? Because this is what it's actually stored for. We said it just a few minutes ago. We talked about it. Major store. Oh, Tanya said, I'm going to go ahead and spell it all the way out. There you go, SR. I sometimes call it SR for short. Sarcoplasmic reticulum. Yes, sarcoplasmic reticulum. Okay? That sarcoplasmic reticulum is storing calcium for this spot right here, that calcium binding site, follow me, on troponin. When calcium binds troponin, it kind of activates it. Troponin pulls tropomyosin off to the side and exposes those active sites on actin so that myosin can attach. Okay? When calcium is present, calcium binds to troponin, which activates troponin, pulls tropomyosin off to the side, exposes the active sites on actin, which is the reason why it was called actin, allowing myosin, not pictured here, to attach to actin. Big picture. Ready for the big picture? What has to be present for muscle to contract? What has to be present in the sarcoplasm, floating around these fibers? What has to be present for muscle to contract? Heba got it right off the bat. Calcium. Yeah. Without calcium present, muscle remains relaxed. Without calcium present, muscle's going to stay relaxed. Okay? Because of the way these 3 proteins are hooked together, well, really, the 2 that are actually attached, troponin to tropomyosin, but tropomyosin is covering actin, the active sites on actin. Myosin wants to attach, can't do it with tropomyosin in the way. Calcium moves troponin, troponin moves tropomyosin out of the way. No calcium, Tropomyosin stays wrapped around actin. Myosin cannot attach. Muscle remains relaxed. Okay? Oh, there we go. If there's no ATP, we will talk about that. I want you to see that nothing so far has required ATP. I'm gonna get to it. I'm gonna get to it. Hold on. Nothing so far has required ATP. Okay? I'm just setting the pieces in place so we can then talk about how they move. All right, so this slide is just a summary of what I just said. Without calcium, myosin cannot bind. With calcium, myosin is able to bind to actin. Calcium is what causes it to contract. Okay? Yes, but I would say that is an oversimplification. I don't want you to memorize it that way. Okay? You do need to know the parts, the sequence of the parts. Calcium binds to troponin. Troponin moves tropomyosin. When tropomyosin moves out of the way, it exposes the active sites so myosin can attach to actin. I think I have a series of illustrations here. Yep. There we go. Okay, we'll get to that. We'll get to that. We'll get to that. I already answered this question. What is the role of calcium during muscle contraction? What does calcium do? And this is where I want you to know very specifically, what exactly does calcium do? Don't big picture. Don't big picture it. Do not give me the big picture. Calcium makes muscle contract. Don't say that. Now, Tanya's on the right track. You're on the right track, sir. But it doesn't bind to tropomyosin. It binds to troponin. And it's troponin that's attached to tropomyosin. So when calcium binds troponin, troponin is already attached to tropomyosin. Calcium binds troponin and it makes troponin bend, and that pulls tropomyosin out of the way so that myosin heads can attach to actin. Yes, that's correct, Melanie. Yep, you kind of finished the story for me. That's correct. Now, what would I accept if this was a short answer question on the test? What is the role of calcium during muscle contraction? I would need for you to say that the key answer is calcium binds troponin, which allows muscle to contract, would be okay. But I want you to make sure that you don't skip to the big picture and just say calcium lets muscle contract. I want you to tell me exactly what it is that calcium binds to. That's the big idea. No, that's the tiny detail. That's the big answer for that one. But we haven't talked about the role of ATP yet, have we? So let's take a look. So, wow, I'm sorry that's blurry. Got these from the textbook. I thought they were high res. So here in this picture, down here in the bottom left, we've got our actin thin filament surrounded above and below by myosin. And they alternate like that. Here's actin. Actin, the active sites on actin are covered with the tropomyosin, blocking the little yellow active sites. So myosin sitting down here, it's ready to attach. It wants to attach, but it cannot because tropomyosin is blocking it. Okay? There's one thing that can move tropomyosin. Okay? It's not calcium. What's the one thing that can directly move tropomyosin? What can move tropomyosin? What can get tropomyosin moved out of the way? let me rephrase it, who is directly attached to tropomyosin that can move tropomyosin, troponin. Yeah. I know y'all want to be talking about ATP, and you know what? I'm going to be talking about ATP. But the reason why I'm holding back on ATP is because I think a lot of students are confused in thinking the role of ATP in contraction is to cause the force of contraction. And the crazy thing is, it isn't. When it's time to talk about ATP, we will talk about ATP, my young Padawans. But until it is time, you are not yet ready to discuss ATP. But when you are ready, I will lead you there. Okay? Let's go back to my question. What's the one thing attached to tropomyosin that can drag its lazy blank out of the way? Troponin. That's it. But guess who else is lazy? Troponin's like, you know what? He looks at you real close and he says, I could move that tropomyosin out of the way for you. I'm going to need something from you first. What does troponin need to get tropomyosin out of the way? Because troponin ain't going to do this unless you give it some what? Calcium. ATP, dang it. Come on. I told you. No, not ATP. Not yet. You're not ready. You're not ready. You're not ready. Yeah, Juanisha, Tanya knows better. You know better than that. Okay, guys, listen up. Troponin will move tropomyosin out of the way, but not in the absence of calcium. Calcium is troponin's payment. It's how you pay troponin to get tropomyosin out of the way. Troponin says, I'll drag that thing out of the way for you, but not without calcium. So you need calcium coming out of the sarcoplasmic reticulum to get that thing moved out of the way. Now take a look right down here in the bottom right. Once it gets moved out of the way, look, calcium binds to troponin, drags tropomyosin out of the way. Now that it's out of the way, look at the difference between this picture over here, where tropomyosin is blocking these little yellow spots, and then we got it over here, we've got it exposed so that myosin can attach. Okay? Now listen to me, everybody. No ATP required. No ATP required. Okay? Does anybody have magnets on their fridge? You got magnets on your fridge? Everybody's got magnets on their fridge. That's what you buy a refrigerator for, to put magnets on. How much energy is required to get a magnet to stick to your fridge? How much energy? None. It's magnetic. It's magnetic. Are you following me? That's what I'm pointing at right here. The connection between actin and myosin is simply a form of magnetism. Requires no energy. Okay? Now watch what's going to happen in my next series of pictures. Now that they're attached, that myosin is going to pivot. It's going to pivot. When it pivots, it will only pivot when it's attached. And it pivots, pulling the actin filament, the thin filament. The whole structure gets pulled a little bit, not very far, but it gets pulled because of that myosin pivot. And again, guys, no ATP is required. In fact, the ADP it had been holding on to, it just lets go of. It releases the ADP. It just lets go of that it had been holding on since the previous cycle. Okay, now watch. Now it pops off and lets go. This is called detachment, where the myosin detaches. Look what's required. You can type it now. Go ahead. You've been practicing. ATP! Yeah! What is ATP required for? The same thing that it takes to get the magnet off the fridge. You don't need energy to get the magnet to stick to the fridge, but you do need a little energy to pull the magnet away from the fridge. So ATP energy is actually required to detach myosin, to detach it from the thin filament. Now, Melanie, yes, this step over here, I was going to show you this in a bigger picture here in a second. This is how the sarcomere contracts Z line to Z line. This is the cross bridge right here. This right here, Heba, is the cross bridge. But what ATP is required for is the detachment and then resetting it. Did you see what I did there? Detach and then reset, which is in this picture over here on the right. The myosin has to detach and then it actually burns the ATP to reset it back into what sometimes is referred to as the cocked position, like cocking the hammer of a pistol. Okay? All right. Hoo-wee. Right? So, wait a minute. We'll get to these questions in a bit as well. We'll come back to all these. I wanted to show that image that I have here on my slides. This is from your textbook also. That's kind of related to what Melanie typed in. Is that how the sarcomere contracts Z line to Z line? So take a look at this image right here. We've got our dark purple myosin and our red actin. And what's happening right here, right in that tiny little spot, and it's happening all along the length of the muscle, is a little myosin head is trying to attach to actin, but it can't because tropomyosin is blocking it. But when calcium comes in, calcium makes troponin move tropomyosin, so myosin can form cross bridges with actin and pull in this cyclic pulling like rowing a boat. But when it does, it doesn't move the myosin, it actually moves the ends of these thin filaments called Z lines. It pulls these Z lines closer to each other. And so you can see from Z line to Z line over here, and then when they pull together, that's what causes the whole muscle to contract. And that span from Z line to Z line, as Melanie typed in her question above, is called a sarcomere. That distance from Z line to Z line is called a sarcomere. It's the contractile unit. So it kind of looks like this. If you have a single muscle fiber at rest and then it contracts, the area where the myosin is stays the same width, right there to right there. Well, my aim is so bad, from there to there. But what actually comes together, that's a Z right there, and that's a Z right there, and those get closer to each other. They get closer to the middle of the myosins, and the space around them kind of contracts. And so the whole thing shortens. But when that same muscle fiber is attached to a bone, then when it contracts, it can pull a bone closer to another bone. That's a bone. So that's how two bones, like your humerus and your radius, that are attached by the biceps brachii, when the fibers of the biceps brachii contract, it pulls one bone closer to the other bone. Let me look at your other questions here a second. Is the cross bridge required ATP? The cross bridge does not require ATP, right? That's what I was saying before. Take a look at this picture here. Here's the cross bridge formation. That's myosin attaching. Does not require ATP. Pulling it off, detachment, is what requires ATP. Melanie's question. So ADP and P comes together to move the myosin heads. ADP and P do not move the myosin heads. No. The myosin heads are moved by the binding of ATP. The myosin head, no, even moving, no. Melanie, I want to make sure I'm understanding your question. This movement of the myosin head that's called the power stroke, that movement of the myosin head called the power stroke, like that, requires none of those. It does not require ATP. It does not require ADP plus phosphate. No. It's a thing that myosin does on its own. It's another one of those magnetic things. It's like a spring. It's like a spring, like a spring-loaded thing. Okay? So this down here, the ATP is required here to get myosin off, and then you have to, the ATPase burns the ATP into an ADP. It separates them, and that is what spring loads the myosin back in this position. Then once it reattaches by magnetism, and then the spring mechanism makes it snap into that pivot. Yeah. No. I understand. The picture be a little bit confusing, but if you guys have a copy of the textbook, this series of 4 pictures is kind of all on one page in the textbook. Does anybody have the textbook handy and can tell me what page this is on? Not the lecture notes, but the textbook. If you've got a page number, you guys can type into the chat. I like these things because at least they are less text than the textbook itself, and the text is divided into like really. It's not clear at first. I'm not saying it's easy. I'm just saying it's more straightforward what's happening in each picture, and it's nicely divided up. Thank you, Dongso. 308, 309. Which edition of the book is that, Dongso? Which edition of the one is that? Is that the 11th edition? Okay. It may be a different page number if you have an older edition. Okay. So what happens to the Z lines during contraction? They get closer together. They move towards each other, Z to Z. What happens to the A band during contraction? Really nothing. I'm going to show you this. The A band is the dark band. Here's what I remember. Here's how I remember it. Watch. I just capitalize that A and remind myself that the A band is the dark band, the dark zone. And the I band is the light zone, the lighter zone. It's got an I in the word light. What happens to the I band? It almost disappears. Okay. What happens to the H zone during contraction? Same. It almost disappears as well. Yeah. The H band. It depends on how much the muscle is contracted. If it's fully contracted, it will disappear. Yes. Yeah. I'm going to show you that picture again. Now we've answered all these questions. Okay. I'm going to do it on this one right here. Okay. Let's label these up. See if I can do this okay. This is the A. That's the dark band. Okay. This is the light band. Okay. There's another one over here. It looks like the A won the gold medal at the Olympics. This looks like the gold, silver, bronze medal. There's the A band surrounded on either side by an I band. There's another A band over here. There's just kind of alternate back and forth, right? There's an A band there. In the middle of the A band is a zone that's a little bit lighter in the middle of the A band. It's called the H zone. All of these were named by the Germans that discovered all of these. The A, if I can remember the word, it's anisotropic, and I is isotropic. It has something to do with the microscope, the way that they did the microscope. But the H one is the funniest one to me because it uses a German word which means slightly lighter, and it's the German word hella. If anybody speaks German, please correct me. I don't want to embarrass myself. This is the way that I heard it, that the word hella is a word that kind of describes twilight just before the sun goes down. It's not really dark and it's not really light. It's kind of in between. And so they described that H zone as the hella zone, the H zone. And I always remembered it by saying it was hella darker or hella lighter, but that was just my way of remembering it, and that was abusing the German language. But in the middle of the I is the Z, okay? So this is relaxed. And then when it contracts, you can see what happens to the A does not change, but the H in the middle practically disappears. The I almost disappears, and the Z's move towards each other. Okay? So those are some kind of key concepts that I want to make sure that you think about as you're studying this stuff over the next week. If you've kind of not gotten to it yet, you're going to be learning this. But if you've already watched the videos and started filling it out, it's a good idea to come back through this again. All right, so take a short break. We've been at it for an hour and a half. We got another hour and a half to go. Well, not quite an hour and a half, hour and 20 minutes to go. And we're going to